and we're still very much engaged with IITians. Okay, so um, wanted to share with people that yes, your dreams can come true of whatever it is that you want to. And I have spoken before to chemistry people, both in the department and otherwise, um, about the fact that chemistry is a very lucrative business to be in, that you can succeed. And I always tell people, look where I got. I went from cutting classes in IIT Bombay to being the MD of a Tata Enterprise. So there must be something right that we did when we were students in IIT Bombay. And so can you. It all depends on you. And I think that if you put your heart to it, you can accomplish any of these things. So, yes, there are career paths in chemistry, all the way from the university to industry, and ultimately the Nobel Prize as well. So, if you really want to do it, you can get there and do it. Um, chemistry is a fundamental science and it's integral to everything in the universe, right? There is no human activity that does not involve chemistry. Life, health, food, and shelter, which is very close to us. And so what? People are thinking, now what do I do though? Okay, I've got into chemistry, I'm doing my BS or MS, and but how do I make a career out of it? So, what do I do next so that I can get personal passion satisfaction? I can satisfy myself for what I want to do, that I'm following my passion. Yeah, but also to ensure a good life. Everybody can't be worrying about passion when there's no khana to eat. So you have to eat well, but you want to also have job satisfaction. It's never enough just to make good money. Many people find that out after making a lot of money, that boy, I wish I had done something different that would be more satisfying. Money is always good, but there is something more. So the chemistry background of where all this is important is, as I said, every walk of life: food science, agriculture, industrial chemistry, polymers, energy, novel materials, academics, even intellectual property and law. Law also requires chemistry quite a bit. Jurisprudence, one third of jurisprudence textbooks are full of chemistry. The one that is closest to my heart is right on top in pink, which is healthcare and medicine. Because if you're not healthy, life pretty much sucks. So you need to be healthy to be able to enjoy it and do all the things that you really want to. It's one thing that will never go away, that need to have good health. And medicine has helped us along in there. So you can see chemistry is everywhere in life. And I'm going to spend time just talking a little bit about healthcare. Now, structure the talk so that I just introduce you to some concepts and some ideas. And then I would actually like to throw it open for questions, because it's more important that you get your question answered, or at least a thought uh, response to some question that you have in your mind to get you going, rather than me lecturing you on chemistry or things like that. Okay, so I'll keep it short. I'll keep the talk short. Introduce you to some concepts of what it is I did and what I can do, and leave it open to questions from y'all. And the questions can be about anything. I've spent some significant time in the chemistry department over the last 15 years, uh, talking to students. And, we have a scholarship from Advinus also in the chemistry department for a couple of kids, at least up until this year. Once I gave up the MD ship, I don't know if they've continued, but we had it for many, many years. And I've spent a lot of time with students answering the kinds of questions that you probably will have. Okay, so medicines are an integral part of good health. Right now, we can't get away from taking headache pills to cancer therapies. Where does all this come from? So originally, aspirin came from willow bark. Everybody knows that story. But people also know that some of the hard drugs also came from natural sources. So natural products and folklore, the trial and error method of seeing if it'll help you feel better. The chemicals that are involved that are available in nature will help and interact with the chemistry going on inside your body and help you to better health. With the structure of aspirin and the isolation of aspirin from willow started the chemical revolution. And so for 50 years, we had a lot of chemicals that were isolated and used for health. Another example is Fleming's penicillin, right? So that is also a chemical, and people isolated that. He isolated that and showed that this could kill bacteria, and that was a revolution. And then came Watson and Crick, and they did their little bit of smoke and mirrors with um, somebody else's data and showed the double helix of the DNA structure. And so they started the biological revolution, the genetics, and how life actually is a lot of chemistry. So when I talk to laymen, I tell them, the idea of a thought in your brain leading to you picking up, say, a book from the table is actually a series of chemical reactions that start from that thought and all the way down to your fingertips to catch the book and up. There are a whole bunch of chemical reactions. It's nothing else but chemical reactions. So we are very much a part of it, and biology is also very much integrated in chemistry. And then, of course, we have now the genetic revolution, which is also very chemical in nature. The latest, of course, is inserting, correcting mutations in genes so that you can get rid of some of these horrible diseases by putting in the right gene. And that's also a series of chemical reactions where bonds are broken and new bonds are formed. And you have, voila, a brand new person with no disease. So these things are all what helped medicine, and this is where it comes from. And where are we going from here? Many directions. Don't want to get into that. It's a very complex situation that involves ethics, law, chemistry, biology, and of course, morality. So it's really a very complex situation, right? Now, how did human beings start thinking about this? What is this chemical and biological revolution? The understanding that life, living things, actually are actually a series of chemical reactions that are carried out by enzymes and proteins has led to the idea of rational drug discovery. That is, if this chemical reaction is not going the right way to produce something that will allow me to pick up the book from the table, what can I do to correct that chemical reaction and make it precise enough for me to pick up that book? That's a rational thought, right? So, identification of molecules that will allow me to do that. That if I create a molecule that can intervene in these chemical processes and change what a biomolecule does, and thereby change the physiology. So allow me to put my hands on the book, pick it up, and read. This is a rational process, and it can be used for a therapeutic effect in any physiological situation that has gone awry, right? What does that mean? Okay, so that means, like, for example, in diabetes, too much sugar running around the blood. Can I do something to reduce that sugar, burn it off, do something to it so it's not running around the blood destroying other things? Yes, there is. There are ways that you can do that, and those are rational ways. They have been uh, taken up in a rational way to get rid of that excess sugar and lead to a therapeutic effect. And that advent of rational drug discovery came after the chemicals that were isolated and found to have biological activity. So, the understanding of biology as a series of chemical reactions and effects of, of chemical reactions led to rational drug discovery and ways to find molecules that can affect biological systems. So, drug discovery, this rational drug discovery, is actually multidisciplinary effort. Right? It has a lot of different things. On top of it is medicinal chemistry, making molecules that can become medicine, molecules that can become medicine. Okay, so it's not just synthesizing any molecules. There's a rationale behind it, and it involves things like pharmacology, pharmacokinetics, toxicology, pharmaceutical analytical R&D, biotechnology, high throughput screening, all these fancy terms. Now, many of these things have chemistry as an integral part of them. And you'll ask, okay, so how does clinical research have chemistry in it? Well, clinical research, you have to understand that the molecules that you make are either in test tubes, they've been tested in test tubes or in animals. In the clinic, you have to understand that the same chemicals must do the same thing in humans. You have to understand that. Are the chemical reactions in humans the same as they are in rats or dogs or monkeys? And it's part of um, that process of translating our chemistry into something that is helpful. So every aspect of this actually is involved with chemistry, but all of it is required to make a successful medicine. You can't make successful medicines without this, and therefore you cannot make successful medicines without chemistry, right? Um, this is more of the same, and this is just broken up into how drug discovery actually occurs. I don't want to go into details, but I can tell you that many of these processes that are part of drug discovery, the steps that you have to take, ultimately involve chemicals. And so chemistry is an integral part of this. Okay. Um, People are nowadays are using protein molecules for treatment, antibodies, enzymes, and such. So where's the chemistry in that? Huge amount of chemistry. The formulation of a protein that you can make and keep it in high concentrations so you can give an injection is not a trivial. It's very, very tough to do that. Also, what do you give it with? How do you stabilize it? How do you deliver it so that it goes to the right place? Can you use that to target where your protein goes? And you use chemistry to do that, and people do that. So many people, for example, make conjugated antibodies. That is, they have antibodies with a poison chemically attached to it that then is injected, it goes to a cancer cell and kills the cancer cell only and not other cells. There also there is chemistry. So emphasis is that chemistry is a very big part of everything. Now this is all technical, right? And it involves pure chemistry. But careers may or may not have only chemistry. They may have other things also. So what do you need to, for success in a chemistry career? Number one, passion. This is really, really important. Um, People who join chemistry just because they got into IIT and are looking, not that interested in chemistry itself, for them to look for a career in chemistry is not important. So whatever it is that you do, one of the organizers was talking about engineering physics. That's her passion. She wants to do that. Wonderful. As long as it's your passion. I told my daughter a long time ago when she was doing pre-med classes, um, that she should follow whatever her passion is rather than do what other immigrants in the US do, go for engineering or medicine. That's not the answer. Because when you have passion for something, you'll excel. And nowadays, whatever you do can be converted into a lucrative career or business or something that will make money for you. Even artists make money ultimately, right? So, passion is the first requirement and thing. You really should want to do this. Then, of course, solid knowledge and education, which most of you are already well on your way with that in IITB. Top and the best school I know. You want to have innovative thinking. Can't be, I was in first year science in Xavier's at the University of Bombay before I joined IIT. And boy, it was like, learning everything by rote. There was nothing innovative at all in what we learned. Nothing, zero, zilch. It was only when we came to IIT Bombay that we were taught what, innovative thinking is, thinking out of the box, thinking of things other than something just learning by heart. I hope that that has continued there. I think it does because many of the faculty have told me that um, they encourage people to think. Uh, so innovative thinking is part of success. Finally, there's knowing what you like doing. Chemistry is pretty vast, there's lots and lots of things to do in any subject. Mechanical engineering also. Many different things you can do in mechanical engineering. Yeah, do you do fluid mechanics? No. Do you want to do strength of materials? Yeah. You got to know what you like doing because once you focus, get to that point, then you can start excelling in that. So knowing what you like doing, the earlier the better, but don't force it. Don't force it. Remember to keep your mind open because you don't know where it is coming, where the next thing is coming from. So you have to keep it open. Finally, patience. Being in science, when I did my PhD, 90% of what we did failed. Now that can be pretty disheartening, but you shouldn't give up. You got to have patience because we are testing the unknown. And that means going down lots of blind alleys and not succeeding. Then you come back and you start again. And actually, science in building hypotheses, finding the answer to that, and seeing if your hypothesis is right or wrong, and then continuing on requires a lot of patience. So you should have patience. Even in your career, you're not going to go from IIT Bombay to being the MD of a 2,000 person strong company. It's not going to happen. You're going to have to go through the various stages, learn different things, and go on. These five things I think are very important in making sure that you prepare yourself for a good career, a successful career. Okay? Now, all this was just words of this, that, and the other, but here's the proof. Let me tell you that most biotech companies, first in the US, a little bit of it now in India, all over Europe, were founded by academics, by professors. They found something, they had an idea, they patented, and they formed a company around. Therefore, what that says is that people have the opportunity to make a financially successful future out of their ideas. The infrastructure is there for us to do that. Okay? It's called patenting and it's called starting up. We did that. We did that when after we quit Ranbaxy, my colleague and I, we formed this company called Advinus Therapeutics. Advinus stands for Advantage, ADV, IN, India, USA, US, because we are American trained people who had come back to India to start this business of innovative drug discovery and pharma research. It was funded by a Tata group. Ratan Tata himself was deeply interested in this. He asked us to come out and make a presentation to him and the board of Tata Sons. We did. He loved it, and we started off this company that went to over 500 scientists, more than half of whom were chemists. Most of them are still there. They're all successful, and we acquired a operational R&D center. So I ended up being an entrepreneur as well, since I was a co-founder of this and a scientist. So I started as the CSO and then went on to be the CEO and MD. But our model was very interesting. We did drug discovery and drug development, the two arms of creating a new medicine. First is discover it, make a prototype, and then convert that prototype into a product through the process of drug development. And we created value by doing collaborative discovery. And we worked with all the biggies in the world: Merck, Novartis, Johnson and Johnson, Celgene, Takeda, and they were all successful collaborations. We made quite a bit of money out of that to help us with the research. In this, we did all the thinking, generated the data, and we patented our things and then gave it to the pharma company. We also did projects on our own internally with no collaboration and no support from large pharma or other companies. This we call internal discovery, where we did our own research, came up with a prototype, and licensed it out. And in fact, right now, I am playing the role of the CSO of a biotech company startup that has licensed our molecules that we discovered um, about 5, 7, 10 years ago. Okay? So they're trying to convert it into products. Finally, in Bangalore, we also did contract services. So here it is pure just money. You have an experiment to do, we do it for you, give you the results, you give us some money for doing it. That's it. And this was our business model in there, and it succeeded very well. It succeeded so well that a couple of years ago, we sold the company to a global CRO called Eurofins, and we formed an internal company with our own intellectual property called Impetus Biosciences. It's also a Tata company. And then we finally licensed out our patents and our molecules to a US based biotech, and have that allowed me to retire. And I'm right now speaking to you from Seattle in the US, where I've retired and I'm having a lot of fun with my grandchildren. So, what I've tried to tell you is that there's lots of opportunity available for all of y'all. There are some key things that are basic that need to be taken care of: passion, good knowledge, good absorption of the knowledge and education that you have, having patience, think innovatively, and go for it. Go for it. Go for what you want to do. Don't wait. Don't think about what, don't wring your hands thinking, what should I do? What should I do? Go and go for it, whatever it is that is your passion. This has played out as I've just showed you by forming a company. And there are many people like me, by the way. From our batch, um, there's another person, Roy Vaz, who has just formed the company. There are people who are working, who are CSOs and big shots in other companies doing research. So, very, very successful careers from chemistry. And one of the things that, many chemists were feeling is like, what do you do here? Chemistry in India, if you go by the old ways, is just not worth it. You have to step out and go out and do something innovative and more modern. And if you do that, success is almost guaranteed. You guys are bright. You all have a lot of uh, goodwill. Many American universities think IIT Bombay, oh yeah, come on in, join in. you know. So y'all can do it, but you need to know what you want to do and what you can do. Okay, having told you this a little bit about this, I'm ready for questions and comments from y'all guys. You can ask anything you want to ask. It can be technical, it can be non-technical, whatever it is that you want, you can ask. Okay, so feel free. Sanjot, I'm ready.
Okay, sir. So I think we have some decent amount of questions here. So we'll start with I think even since number of people is quite low, we can even actually ask them to unmute and basically unmute and do that. Roshni, can we do that?
Yeah, sure. We can go ahead with that. I'll just make sure they're
Okay,
unmuted.
so Arvind has asked a question. So can you unmute Arvind and ask whether I think you can let him ask himself.
Yeah, sure. Yeah, Arvind, you can go ahead.
Arvind, you can actually speak. Okay, he has trouble. Okay, I think until then we can let Alok unmute and ask. That's fine.
Yeah, Alok, you've been unmuted. You can go ahead.
Okay. So, hello, sir. Thank you for the talk. I was interested in knowing that, firstly, how did you identify the market wherein you wanted to launch your new company? And like, what knowledge from your previous working in the professional domain or in the academic domain helped you identify that knowledge gap? And could you explain your journey of finding that niche and developing your new company?
Sure. Um, it's an interesting thing. I was doing very well at a global company called Bristol Myers Squibb in Princeton. I was doing very well there. I was highly thought of. When a colleague asked me if I wanted to return to India to head up drug discovery, and I thought to myself, and I thought about it for a year. I looked at what was going on in India, and here's what I found: innovation in pharma research in India was zero. There was no real drug discovery going on. Most of it was generic and copycat medicine because the patent regime in India was still what Indira Gandhi made it in the 70s, that there was no way to patent any discovery or invention you made. Okay? But in 2005, India was going to sign the TRIPS agreement, where if you had an innovative idea, you could patent it, and India would honor that patent exactly like the rest of the world. So this gave incentive to people in India to be able to cash in on the ideas if they had any. That's one. Two, Ranbaxy at that time was the largest pharmaceutical company in India, and the owner, Mr. Dr. Parminder Singh, had hired Davinder Brar and asked him to convert it into a innovation company. That is, a company that would do research, find new medicines, and convert them into products. Okay, so he was interested in doing this drug discovery business, and India was ready for this new pharma research. Why? India had the same infrastructure of education in chemistry and biology as the Western world. China did not. China didn't have that kind of infrastructure. So we had the masters and PhD programs and all this stuff. We taught in the same way. And India was ready for this new sunrise industry after IT. That new sunrise industry was going to be innovative pharma research. So we actually were the first people to come back in what is now called the reverse brain drain. And actually, if you Google me, you'll find a couple of major newspaper articles and magazine articles on us coming back to India to start this. So an opportunity existed in India to use the talent in India to do innovative pharma research. I actually visited Ranbaxy 3 or 4 times over that 1 year I was thinking, and I talked to the scientists. And I thought the scientists there were superb. They were very well educated, they were passionate, they had the fire in the belly, and they wanted to do things. And I thought, boy, yeah, if I can share my experience with them and just show them the ropes of how to do this, it ought to be pretty successful. And it turned out to be true because in 9 years they had made 2 prototype molecules. In the 2 years that I was with Ranbaxy, they made 9. So 9 years, 2 molecules, and 2 years, 9 molecules. That's how good that team was. So the talent existed. We joined the TRIPS agreement, and now we were ready to do pharma research going from generics to innovative pharma. All of these things played a part in trying to create this in India. Okay?
I have a follow-up question. When you sold your company, what were broadly the knowledge and the products that you transferred, and what were the things that you retained? As you said, some patents were retained, and uh, so how does that work out?
Yeah, okay. So when we sold the company, uh, there was a global CRO, contract research organization, that did a lot of lab work. They wanted to buy our contract services, which was the larger part of the organization. Remember, drug discovery is usually a much smaller organization. So we had about 200 scientists in there, and we had 500 scientists in contract services. So they wanted to buy the contract services. We wanted to sell it, and Tata's wanted to sell it because Cyrus wanted to get out of this innovation pharma R&D business and stick to core businesses. So Eurofins agreed to buy the contract services. They agreed to buy the whole company, but we told them no. Our patents that we discovered, the new molecules that we want them back. So we bought it back. We sold them the whole company and bought back our own patents and kept it, and formed a new company called Impetus Biosciences as a holding company for those patents. So those patents are for new medicines that will one day hopefully become medicines. Okay, and of those we have licensed out 4 of them to a biotech company here. They are prototypes. They are chemical molecules that have been identified, and they have now to be tested in humans to see if they work and are safe in humans before they become products. Most of the failure comes in testing in humans because you can't do experiments on humans. No? So that has to be still done, and the process has started. So all our internal patents, all the patents that belong to us, we kept in the new company, and we are out licensed out 4 of them. In our collaborative discovery, we actually have 2 products, 2 prototypes that we have given to Takeda, and one we have given to Novartis. This was done in collaboration with Novartis, and they are actually taking it forward for um, fatty liver disease, and it's now in phase 2B trials. So very successful. But the patent belongs to them because they paid for the research, and they will give us royalties as well. Okay, so that was a collaborative discovery. The patent belongs to them. The internal stuff that we did on our own, we kept and are licensing it out.
Okay, Alok,
Thank you for the detailed answer, sir.
okay. So, for next question, I would like to ask Arvind. By the way, just a announcement. We have floated a feedback form. Please do fill that and let us know. Roshni, can you unmute Arvind? I think he is already unmuted.
I think, yeah, can you hear me?
Yeah.
Yes, yes.
So yeah, I had like a three-pronged question. They're all fairly long-winded, so I'll try to keep it short. I won't read it out. So in context of this is technical. So in context of the superbugs that we hear about, MRSA, extremely drug-resistant TB, and all that stuff, I keep hearing that we need to find an entirely novel class of antibiotics. So in that context, like what defines a class of antibiotics, and what does moving away mean? Do we have to move away from like conventional alkaloids that we've been using? You showed us a diagram where you said naturally derived substances and synthetically manufactured substances. So I'm just wondering what moving away means and what defines a class. That's one. I can read out all of them, or we can go one by one.
Let's go one by one because then I'll get confused and not answer your question as precisely as I would like to. So,
Sure.
classes of molecules in anti infectives or antibacterials is based on their mechanism of action. So far, they have all been derived from natural sources. They are a product of finding natural products that have shown antibacterial activity. If you remember, penicillin was discovered, penicillin was made by a mold that killed bacteria, and it was discovered by Fleming that way, right? So it actually inhibits cell wall synthesis. And so the class of molecules is cell wall synthesis inhibitors. Okay. Then another class of molecule, if you look at the mechanism of action, is translational inhibitors. So tetracycline, those kinds of molecules inhibit protein translation in the bacteria. So they are ribosome inhibitors. Okay. The third class of molecules are uh, they're called pump inhibitors. So bacteria have these protein molecules sitting on the cell wall that if a foreign chemical comes inside it, it pumps it out quickly before it can do any damage. Okay. So if you find a molecule that actually inhibits that pump, then the toxic molecule stays inside the cell and kills the cell. So the classes of molecules are based on the mechanism that they work with in a bacteria. Many of them are uh, derived from natural products. They are found from nature and then optimized for better suitability to humans, either in the toxicology or in what we call pharmacokinetics. Okay? Um, we have to move away from that is because there are too many resistant strains that are cropping up. Now, how does that happen? Resistance to these drugs comes via what are called plasmids, which are DNA molecules that enter the bacteria and produce a protein that neutralizes the drug. Okay, so resistance, one of the most common resistance proteins is the resistance strains is these pump inhibitors. So if you find something that allows the pump to pump out the toxic molecule, you have resistance. Now, we need new mechanisms because the bacteria has not reached the stage where it can build resistance to that just yet. It's the overusage and the years and years of usage of all these other drugs that has allowed bacteria to develop resistance to this. New mechanisms will take a long time for that to happen. That's why we need to get away from that. Why are we not there yet? A couple of reasons. One is that um, looking at natural sources has kind of become less fashionable, which I believe is actually a mistake because I think nature can teach us a lot more than we can figure out ourselves. It's one. The second is that in the Western world, where the large markets are, infectious diseases, bacteria are not that common. So there's not that much money to be made by these drug companies. And so what these things are called they're called neglected diseases. They're neglected by the drug companies because there's not that much money to be made, and so they don't do that much research in it. There is some resurgence of this search for better antibacterials, but it's still not taken that much hold where people are pouring enough money into research to come up with new compounds. Okay? So a new mechanism has to be found, enough money has to go into it, and then these molecules have to be developed to be safe and effective. That still needs to be done. Okay? Answer your question?
Yep. Thank you. And I'll just ask you one more from me.
Yeah.
In relation to SARS-CoV-2 or COVID-19, I read something about CRISPR being employed as a potential therapeutic, like injecting CRISPR and having it target the pathogenic genome. Is that as close to a silver bullet for pretty much any kind of infection that we're going to get to, or is it pretty limited?
See, CRISPR is a fantastic technology, but its performance and its safety in humans is completely unknown. It's all right to do it in mice, and it's all right to do it in bacteria and ducks. But if the duck dies, you just can get another duck. Or if a mouse dies, you just get 5 other mice and do it on them. You can't do that in humans. So there's no safety established. There's no safe way of doing it. That's one. Second is, in mouse, we know how to deliver it to the liver or to the muscle or to the eye. We don't know how to do that in humans. You can only inject it into the blood and hope like hell that it goes to the liver or the eye or wherever, right? Goes to the right cells where the excision and the insertion of the correct gene is involved. Those are all still very, very much in humans, untried, untested, and unknown. So for COVID-19, we don't even know how it gets inserted, if it gets inserted, and how long it stays there. It is such a new phenomenon that there's very little known about it. Now, let me tell you one thing right off the bat. There is nothing that kills viruses. The virus itself is a non living thing. It only lives when it's inside a human cell. That's when it's alive. Otherwise, it's just a package of uh, DNA, RNA with proteins. That's it. It's just a package. It doesn't live by itself. It doesn't do anything. It can't move. It can't eat. It can't do anything while it's outside a cell. When it goes inside a cell, the virus uses the machinery of that cell to reproduce itself. And it's not as if it takes out a sword and starts, bullying the cell into doing nothing. It just, the DNA is sitting there, the inside the cell doesn't know any difference. It just uses that DNA and makes more DNA and more RNA. And that's how it does it. So there's no way to kill a virus like that. You can't kill a virus. You have to stop its reproduction inside the cell or stop it from getting inside the cell itself. Okay? So COVID-19 is not going to go anywhere till we find a way for it to stop infecting the cells that it does. And once it does infect, how to stop it from reproducing. Okay? That is one way that people use for vaccination against all viruses that we have: smallpox, chickenpox, those triple vaccines. All those vaccines work by creating an antibody that recognizes the virus particle or cells that have the virus particle and killing those cells. Okay?
Yep, just to
So it
clarify,
does not, it doesn't do anything to the virus per se. It just kills the cell that has the virus in it, so it can't reproduce. That's how And that's how antiviral medicines work. Now, some antiviral medicines work because they get into the cell and stop the replication of the viral genome, DNA or RNA, depending. So you have reverse transcriptase inhibitors for HIV, which will inhibit the reverse transcriptase of the virus. So it can't make more copies of its RNA, right? Therefore, you cannot make more virus particles. But there's nothing like antibacterial to kill a living cell. A bacteria is a living cell. It can reproduce, it eats, it sleeps, it moves around, it reproduces, it does everything on itself. So you can kill it. It's a living thing. Virus is not living till it gets inside a cell. Okay?
Just a clarification that I think the CRISPR was designed to actually target the
You'll have to come closer
VNTR
to the
in the.
mic. I can't hear you.
Sorry. I think yeah, the CRISPR thing was just. I think the approach they adopted was to target only the VNTRs in the COVID genome sequence and not the parts that were in the human genome. So I think they want to just cut it up and then leave it in a kind of useless state whereby even if the cellular machinery reproduce the
Yes,
code,
you
then it won't do
can.
that.
Yes, you can do
But yeah,
that. But how do you get it to the cells that have?
sure, yeah,
You can't,
I
right?
understood
How
that
do you
point.
do it?
Yeah. Thank you.
Okay, I think we next up is Rohit. Roshni, can you unmute Rohit and let him ask?
Rohit, you've been unmuted. You can go ahead.
Thank you. Hello, sir. Yeah, the talk was very nice and enjoyed it. My question is, you must have met a lot of people or startups in India. So I wanted to know where do they fail or what are the crucial challenges they fail to succeed upon in the like field of medical equipments or healthcare, invasive, non-invasive technologies in the all these things.
So, important lesson for all of us to learn is the following. You may have a great technology, you may have great idea, but that does not lead to a successful product. So, conversion of an idea to a successful product requires two or three things that are absolutely critical. First is there a market for it, right? I mean, you may invent a fantastic, I don't know, rocket ship. But how many people are going to buy rocket ships from you? Three, right? So it has to be something that there has to be a market for. There have been many failures just because I was assessing a um, a machine that could I said that could identify um, healthy sperms from non-healthy sperms to increase the chances of fertilization and better child for couples who did not get children. But there was no market for it because couples just said, "Hey, we'll keep trying till the healthy sperm does its job, and we'll have a child. Why do we need to have this machine and go through this whole process, pay a lot of money to do this when we can just have fun every night?" So it has to have some value. It has to have some market that you can go to. That's one thing. The second thing is that converting an idea into a product also requires passing through regulatory barriers. What does that mean? So the government and society in general has certain rules and regulations about what you can do and can't do. Okay. Now, I told you that I have licensed out some prototype molecules for cancer treatment. And it took us 3 years to discover them, and it's going to take 12 years to convert them into a product in which they may fail. those 12 years of testing are dictated by society and by the government. You have to do it this way, you have to do it this way, it has to be safe, you can't just, play around with human lives, such, so on and so forth. So that conversion of an idea and a prototype into a product requires this business of regulation, passing through regulation. And finally, when you're doing it in the lab and you're making 3 prototypes, it's easy to do. But now, when you're thinking about making 320 million prototypes, it's a totally different problem, right? So, a whole new thing has to be developed to convert that and make it into a product. Many people, many startups in India had not thought it through and are fixated on the ideas. It's interesting. I gave a talk on innovation at the Golden Jubilee thing, and this question was discussed in actually quite in detail. It's very hard for inventors and innovative people to give up on their ideas. They think that I had the idea, so I should be able to convert it into a product. No, it's not true. You need other people with other expertises to look at that and tell you whether this is going to be good or not. It has to be commercially viable. If we go back to my slide, yeah, I'll just show you. You see in this diagram, there's down here commercial input. This commercial input is telling you whether there is a market or not in this. It is going to be a viable product or not. Regulatory is going to pass through all the process that the government puts in place so that everything is safe. Process R&D is converting from making 3 grams of molecule to 3 tons of molecule. You see? So it takes totally different things. A startup may have a good idea and start coming up with the prototype, but converting the prototype that is commercially viable, can be developed in clinic, and become successful in the marketplace is totally different. And many people fail because of that. Okay?
Okay, sir. Thank you.
For next question, we have Valay.
Hello, sir. My in this commercial race of um, getting medicines out as soon as possible and then undergoing the process of patenting them and having the exclusive rights, where does open source movement stand here? As in, like open publishing, open use, and that
Yeah.
kind of stuff. Because while patenting is actually helping people to develop uh, like develop science economically, it inhibits people who lack as much resources to do so. So
Yeah, good
what's
question.
your opinion?
That's a good question, and it's always an iffy thing. When I used to speak to the government of India about these aspects, and where drug discovery and open source, and Mashelkar had started a larger effort on this open source innovation. Um, there are places for it, and the place for it is in the early part of it. The generation of the idea and the research that can lead to the prototype can be very much open source. And in fact, in the current situation, it is open source. Academic people do their research and most of them publish it. And for them, the reward and the satisfaction is in the publication of this new idea, new pathway, new way of doing things. Taking that idea and then applying it, making it an applied effort, to create the new medicine, there is a smaller role of open innovation. For example, I can tell you that there are many sites that if I want to make a particular molecule and I'm having a hard time trying to make it, I put it on the open site and ask help from anybody out there. And it has worked for some people. People send in their solutions to the problem, and anybody can, and they get rewarded somewhat, a little bit, to keep them interested and engaged. And the person with the problem has a solution. So it is happening for chemistry, it is happening in chemical pathways. For biology, it is happening in identification of novel pathways and branches or targets. So there is open innovation going on a smaller scale. But ultimately, when you're making a product, the idea is to make some money. Otherwise, what is the incentive? The biggest driving factor in 21st century is paisa, right? Everybody wants to make a lot of money. So till you don't have the incentive, why would you do anything more? Some people do it, most people don't. Okay, so it's happening, but at a lower level.
Okay, thank you.
There are many sites. If you're interested, you can just Google it. There are many open research sites where you can also participate. You can register with them. You have to have some credentials. Anybody can't just register. So if you tell them you're a chemistry student or a mechanical engineering student, then they, if you become a member of the site, you can provide your solutions as well. There are many such sites.
Okay, thank you. Next up, we have Siddhant. Siddhant, can you, wait, Roshni, can you unmute? Yeah, Siddhant.
Yes, sir. Thank you for the talk. It was quite interesting. Uh, as an undergraduate first year chemist, I've been quite interested, and I've been hearing these topics like computational and otherwise drug discovery and personalized based medicine. Would you share some thoughts on how one should start, or know whether I'm passionate about such related fields, and how should one just go on this route and explore these fields?
Um, I would not make any decision till you actually do a little bit in the fields that interest you. Okay, so I'll give the example of what my daughter. Um, in her first year of college, she started taking pre-med courses, and I told her that she can do whatever she wants. She doesn't have to ask me for any permission or anything like that. When I saw a report card, I noticed that it was all pre-med, and I said, why are you doing all pre-med? Is that what you want to do? You want to be a doctor? And she said, yeah, all my friends are going to be engineers and doctors, so I thought I'll do it too. I said, but is that what you want to do? Is that your passion? And so the advice I gave her, I said, why don't you take an extra year and go and try out all these different things, art, business, take courses in that and see what really turns you on, what makes you want to wake up and start doing that stuff every day. And so she did that, and she ended up doing business administration and art history instead of medicine. So that was where her passion was, and that's what she wanted to do. Now, she did that because she actually went out and took courses in that to see if it tickled her fancy, which is what you would have to do also. Now, you have to understand that there are different um, modalities to the kinds of things that you are going to be faced with. You named computational. You also named biotech. I was advising the India, the head of the National Institute of Bioinformatics. He came to my office in Delhi and was asking me, how can I get more people and the right people to join my course in bioinformatics? So he said, I asked him, where are you getting your students from? He said, I've got a whole bunch of computer science graduates who are doing this, who are joining us. But, they're not doing that well and they don't stay in it. So I said, okay, well, stop and think what you're doing. You're doing bioinformatics. The questions you are asking in bioinformatics are biological in nature. The process you're using is computational in nature. If you get computer scientists, you have the process in place, but you have no questions to ask. How do you generate those questions? I told him I think it would be a better idea if he went and looked for biologists or chemists who are interested in finding computational solutions to the questions that they have. Do it computationally rather than do it in the test tube or in a mouse. Can you do it computationally? And he started doing that and it was more successful. So my thing to you is the same thing, is experience these aspects and see what turns you on. Let me tell you my own story. That'll maybe help you a little bit. So I was in IIT. I was in organic chemistry, synthetic organic chemistry. I really liked it, and I liked all this, reaction mechanisms and stuff like that. A great professor called Arvind Mehta, who was from Oxford, an old time guy. He was very old when I was in 75. He used to teach organic reaction medicine. I used to love all that electron pushing and all this stuff. I took a course in enzymology taught by uh, Anil Lala. I think he's also retired now. No, I think he passed away. Yeah, I think he passed away. Anyway, he taught a course in enzymology where he showed enzyme reaction mechanisms, and the mechanism he showed them as organic reactions with electron pushing. The one he showed at that time was the serine protease charge triad. I found it so fascinating that I wanted to do enzymology only for the rest of my life. It really clicked in my head like nobody's business. And I have that book today. I still have that notebook with that page in which the reaction mechanism is shown. Why that happened? I took the course. It was a PhD level course, and I was doing my master's that time. I took that course and exposed myself to it, and I found that this was fascinating. It was aligned with what I liked, which is organic reaction mechanism. So you have to actually go out and do it and see what turns you on. You want to see where you're putting in extra work. You want to follow up more and see what it is that is really exciting you. That is where your passion will lie. Okay, that'll guide you towards your passion. Once you have decided what it is that you think, which is on my slide, right? But the fourth point on my slide of what next is knowing what you want to do. Do you want to do computational chemistry? Do you want to do synthetic organic chemistry, create new molecules out of simpler stuff? Do you want to do biochemistry? You have to figure that out and see what turns you on. And then put your head down to it and go to it. Okay? You won't get it done unless you actually do something in it. Actually, IIT Bombay gives you good opportunity. I remember we had all kinds of fabulous courses. Drugs and biologically active compounds. That was one course. No? Genetics was another course. Enzymology, I told you, enzymes and coenzymes. And it gives you enough opportunities to think about and experience other things that tell you whether this is your cup of tea or not. Okay?
Yeah, thank you, sir. That was pretty good.
I think next up we have Kedar.
Yeah, hello, sir. Thank you for the great talk. So, my question is that in any developmental stage of a drug, it has like multiple stages of it. Like first you have test tube trials and then animal trials. So, in what way does the process for human trials differ or stay the same for other kinds of trials?
No. So, one thing is very, very different, and that is that human trials are highly, highly, highly regulated. Everything about a human trial requires multiple layers of permissions, multiple clearances of protocols of what to do, multiple layers of assurance that things are pure and clear, and multiple levels of secrecy. That is, you can't cheat. No, cheating is not allowed. For example, all data is locked. The investigators who are doing the trials cannot see what the data is till a third party who is not related to any of this stuff unlocks the data after it is generated and puts it in order. Then only you can see it. So it's a very regulated thing. That's one thing. The second thing is that it has to be very simple. You can do a lot of manipulations and a lot of things in animals. In the test tube, you can do anything essentially. In animals, there are some rules so that you're not cruel to animals and you have to treat them with respect and kindness. But you can try a lot of experimental stuff on animals. In humans, you cannot try experimental stuff. You have to do it in a very organized. So let me give you an example. I have discovered a molecule which is an enzyme inhibitor, right? It is a nanomolar inhibitor. And what do I do to test it in animals? I first do what is called a pharmacokinetic study. That is, I inject it or give it orally and see if it reaches the blood and reaches the tissues that is there. If it reaches the blood and stays there for some time, I say, okay, I can test it. Then I take a mouse with diabetes and give him some of this drug. And there's no limit that I can only give him little or more. I give him any amount that I think is reasonable and test it and see if it works, right? The animal dies, it dies. Okay, so it's toxic. In humans, you have to start with the lowest dose first that has no effect. You have to wait and see if that has any kind of effect on the patient, side effect on the patient. Then you go to the next higher dose. Then you go to the next higher dose. Then you go to the next higher dose. And the doses have to be at least tenfold less than what had shown toxicity in two different species of animals. One small animal like a rat mouse, and one large animal like a dog or monkey or something. You have to go at least tenfold lower than that is the highest dose you can go to in humans. So human trials are very focused, very regulated, and they have to be ultra safe for the patients. You cannot just do anything you want to, like you can do in a test tube or like in animal studies. It's a very, very controlled effort. Okay?
Yeah, thank you, sir.
I think next up we have Roshni herself. So Roshni, you can ask.
Yes, so sir, I think you've already answered the first part of my question. Was that things like data analysis and machine learning are increasingly being used in as tools in all fields of research? Would you need to have a strong background in biology to move forward with careers like yours? And a second part of this question I have is, what about biophysics? Would there be openings for biophysicists in fields like drug discovery and development?
Absolutely, absolutely, absolutely. Where a lot. In fact, I can tell you that computational chemistry, biophysics, uh, docking, then folding of proteins. So many proteins, the structure is not known. Okay, so to fold it and do the energy minimizations there to come up with a structure that has a chance of working, it's a big effort. It's a very big effort. Also, another very important part of biophysics, my degree, my PhD was from the Institute of Molecular Biophysics, actually. So there were lots of very interesting projects that were going on in there. Yeah, there is ample scope for biophysics as well. The thing you have to catch on is that you have to create the opportunity. One, two, you have to also scope out what is interesting people. What are the needs that people are facing that can be answered by biophysically? That's the key. So remember, I told you I just gave the example of bioinformatics, and there are all these computational scientists trying to answer biological questions. Well, they don't know the question. So question is the most important thing in science, right? Then you design the experiments to find out whether it works or not. There's lots of scope in biophysics too.
Yes, thank you, sir.
Okay, I think we are nearing the end of this session, and don't see any new questions. So I think we can wrap this up. I thank Kasim sir for taking time out of his schedule, even if it's like 11:30, almost 11:30 for him right now. And I also thank all the participants for joining. Uh,
Great.
so
Thank you.
yeah.
My regards to everybody there. Okay.
So thank you once again. We hope you to see in the next sessions. Thank you,
Okay.
everyone. Goodbye.
Okay. Thank you. Good night.
Good night, sir.